So I would like to introduce you to a very wonderful man. This is Stefan Kurtenbach. He's with Bascom Palmer. He's one of their top researchers. He works hand in hand with Dr. Harber. This is the person who is researching all of our genetic abnormalities and working on some really exciting things. So Stefan's going to give you a presentation of what he's got going on. Please feel free to ask any questions after he's done, and if you have any questions, please see me after this. Yes, sir.
Would we get a copy of the presentation?
Can we get a copy of the presentation?
I think so.
Yes, we'll try. Okay, thank you. Here you go, Stefan. Thank you so much.
Well, thank you, Melody. Good morning, everybody. Good morning. So I'm a scientist in Dr. Harber's lab at Bascom Palmer in University of Miami. And I'm going to give you a brief update on the research, on the stuff we've been working on. And I apologize in advance. Some of this is a little convoluted. I tried my best to keep it simple, but feel free to ask questions during or after. So we're located in the Biomedical Research Building. And if you look over here, you can see the sign of Bascom Palmer. So it's literally 2 minutes from there. So together with the Sylvester Comprehensive Cancer Center nearby, we're in an ideal spot to study uveal melanoma because we can go into the OR, get a fresh sample, go to the lab, and without freezing or anything else, look at it. So I'm sure you've seen this before, but just as a brief introduction, uveal melanoma is derived from uveal melanocytes, which sit in the iris, the ciliary body, or the choroid. It's the most common eye cancer, and it's like 5% of all melanomas. We have around 2,000 new cases per year. And half of the patients develop metastasis, unfortunately. For the primary tumor in the eye, there's a couple of treatment approaches. Some common ones highlighted here. We have radiotherapy or enucleation. You can see an example of a plaque up here where 3D simulation helps us to figure out how to place it and how much radiation to put on. And with all these things, we get a pretty good control of the primary tumor in the eye. So this graph up here shows how many people show growth of the uveal melanoma after it's been treated, and it's really, most of them stay clear. An example over here, 2 years after radiation, the tumor stays nice and small, the blood vessels have retracted. So the primary tumor is not the problem. What we see, unfortunately, that even if we remove the primary tumor, like metastases develop in around half of the patient. And in most of the cases, the liver is involved. So despite better treatment for the primary tumor, we're still having a lot of struggles finding a treatment for those metastases. Now, if you look at that graph over here of the metastasis rate, you see it's not a clear line. It's more like divided into 3 different sections, right? So if we take uveal melanoma tumors and we sequence them to figure out what the gene expression in them is, and use a smart computer program to figure out how do those cluster clearly into different areas. So you see one over here, which we call class 2. It's very different from the class 1, and the class 1b is up here. And again, if you link that to a metastasis rate, you see the class 1a has the lowest chance, the class 1b somewhere in the middle, and class 2 the most metastasis. This rate of metastasis also correlates with the mutations we find in the genome, where F1AX mutations are associated with a low metastasis rate and BAP1 mutations with higher. So when I started in the lab, we really want to figure out what these mutations do. We know they're there, but nobody really understands what they do to the cell that cause it to develop a tumor and develop metastasis. And in particular, we're working on 3 things, or my projects involve looking at gene AQ and gene 11, BAP1, and PRAME. So today I'd like to show you 3 research branches in our lab that we're very hopeful about and think that will impact how we treat uveal melanoma in future and hopefully make it a less severe problem than it's right now. So this overview, I want to show you a little overview over how the genome is composed and what BAP1 is actually doing. So we all know we have chromosomes, and if you zoom down in there, we have those DNA strands wrapped around what we call histones. And this the cell decides whether a gene is expressed or not by putting different modifications on these histones, despite many other things. Every year that's passing right now, we're gaining more insights into what happens to a cell that changes what it does. It could be three-dimensional structure of this. It could be methylation of the DNA. It could be how these things are accessible and winded. And also, what chemical groups are attached to these things. And one of these chemical groups is ubiquitin. If ubiquitin is attached to these things, that will shut down gene expression. Now, BAP1 is known to come in and cleave off ubiquitin. And we can see that in cell culture in the lab. If we take normal cells and we get rid of BAP1, we see all the ubiquitin starting glowing here in the nucleus, showing this is indeed what BAP1 does. A second modification I want to show to you is the acetylation, which does the opposite. It doesn't repress gene expression, it activates it. And we all have heard about HDACs. HDACs come in and chew off this little thing, and HDAC inhibitors are the ones that inhibit HDACs from getting that off. So enough for this. So to look into all of these complicated mechanisms and see how can we exploit them for drug treatments, we need good models. And one of the models we're proud about and developed in the lab is cultivating normal human uveal melanocytes. That is something that is not readily available. But we're fortunate to have the OR so close, so during enucleation, we can go in, take normal melanocytes and try culturing them. And we have succeeded. And we developed this model where we can now add a drug and cause all the genetic changes we see in tumors happening. So we can knock down BAP1, we can express PRAME, we can put in gene AQ or gene 11 mutations and see what happens to a normal cell when we put in these mutations found in the tumors. So first, let's look at the ubiquitin, right? What we see here is the whole genome, every single gene lined up in an experiment that's called ChIP-seq for ubiquitinated histones. And as we expect, if we knock down BAP1, that ubiquitination goes up. So the first idea was, well, those genes, those should all be repressed. We have a whole lot of ubiquitin on there. Maybe that's what BAP1 is doing, but it's not that easy. So down here you see different cell lines, and we knock down BAP1 in every single one of them, and this is looking at tumors. There's not more genes repressed. There's the same amount of genes going up than down. So it's a little more complicated than we thought in the beginning, and there's something going on. But what is interesting is if you overlap all that data and ask, what is the common thing here? Like, what is BAP1 doing? What we're finding, all the genes that go up are immune-related. And we think what they are doing is they're shutting down the immune system, making it dormant, and preventing it from attacking the tumor. Whereas all the genes that go down could be other targets. Now, how do we get more insight into this? What you see here is a single-cell RNA sequencing experiment. Now, what people have done in the past to analyze uveal melanoma tumors and others is taking a chunk of tumor, mincing it up, and doing whatever experiment they want to do with it. The problem with that is that you have a lot of other stuff in there, too. You have immune cells, blood cells, normal tissue, dying cells. So it's really hard to figure out what exactly comes from the tumor and what comes from somewhere else. This is a novel technology where you mince the tumor up and take the single cells and sequence every single one of them. This is very fresh data out of the press. We haven't published that yet. This is $100,000 worth of data. It is 50,000 uveal melanoma cells derived from 10 patients. And this is basically the same plot I showed you before with the class 1, the class 2, and the class 1A, and so on, just in a little more complicated and a little more colorful. But we can cluster all these different cell types now here. And we can ask specifically, what immune cells do we see? What immune cells do we see in PRAME-high tumors? What immune cells are missing when BAP1 is missing? So this leads us to develop novel therapies and ask what could we apply to make the immune system here more efficient. So this is the first branch we think is very promising, enhancing the immune system, preventing the tumor from shutting it down. Second, let's go back to the ChIP-seq data once more. What you see down here is one gene, one single gene singled out, and you see the gene expression. And all the red stuff here shows you that this gene expression is shut down when we knock down BAP1. So what happens? The biggest change we find is in this little acetyl group I showed you before that is modified by HDACs. It's not the ubiquitin. It's not any of the other marks. It is the acetyl, which is interesting because we know that HDAC inhibitors are among the most promising drugs we have so far. And if we put an HDAC inhibitor on a class 2 cell, we published that a couple years ago, we can shift the class 2 signature towards a class 1 signature. And we also showed in a mouse model that HDAC inhibitors in principle could work. The problem with HDACs are that there's not only one HDAC we can inhibit. There's like a dozen or so. And this doesn't include splice variants or modifications of those HDAC inhibitors. All these drugs target a different subset of that to a certain extent. And of course, you don't want to use something very broad if you don't have to, because that has bigger side effects. You want to use something very specific in as high dose as possible to target this. But nobody knows which HDAC is really important in uveal melanoma, right? We think, besides the known things, BAP1 interacts with a lot of other things, including HDACs, but only very briefly, so it's hard to detect. To detect these brief interactions, which could lead, for instance, for BAP1 cutting off ubiquitin of an HDAC, we use a trick. We put a little biotin ligase on top of BAP1, which then will put a little biotin group on all these targets that only are touched very briefly by BAP1, and we can enrich for those and look at them. And indeed, what we see is that a specific set of HDACs are in close contact with BAP1, and we think those are the ones we need to inhibit in future. So what do we do if we detect some targets we want to inhibit? The first thing is we go to cell culture. We have a huge library of uveal melanoma cells with all sorts of genetic changes, and we can test it. We can put some drugs on there and just simply ask, at which concentration and which drug kills the cells. A second screening approach is a little more unusual. It's using an African claw frog. It's this big frog species. And people take the oocytes, and you can just develop them in a petri dish to tadpoles. And when you take the oocytes out, you can knock down BAP1 in half of them, and look what happens to the tadpole. And as you can see on the side that didn't have BAP1, this tadpole is missing an eye. So this is very interesting from looking at the developmental standpoint, understanding the functions of BAP1, because this system is more complex than a cell culture. But it also can be used for screening, and people do that, where you just have a huge batch of oocytes, treat half of them, and see which ones grow 2 eyes, which one grows 1, to put it simple, to have a more complex system. The third one is our mouse model. Does this work? Let me play this for you. What you're looking at here is a high-resolution, 3-dimensional ultrasound analysis of a uveal melanoma tumor growing in a mouse. This is a really neat technique to monitor tumor growth in a mouse, to look at how the therapy is working, to calculate the volume of the tumor and how it's behaving. And indeed, putting on some of our new HDAC drugs, we see very nice success rates. So in blue, you can see the animals that were treated with the HDAC inhibitor, and the control animals that haven't received any treatment after a couple of weeks develop these tumors. You can see the date. It's all very recent data, and we're still working on it, but these results are very promising already. So this is the second big research branch leading to therapies, hopefully, looking for more specific HDAC inhibitors. For the third one, I'd like to talk a little about PRAME. Recently, or a couple years ago, we found that PRAME is associated with higher metastasis rate in class 1 tumors that generally have a lower rate. And again, this clustering shows you that if you look at all the cases that develop metastasis versus those that did not, there's a clear difference. And you see one of these circus plots around here, that's the whole genome, and red means this gene is up, and blue means it's down. The most striking change in those tumors is PRAME. We even had to cut this off a little here because it wouldn't fit in here. So that is the hallmark change for metastasis in the class 1 tumors. And this is the most recent picture we have published on what we think is happening, including PRAME now. So where you start off with a normal melanocyte, we then gain a gene AQ11 mutation, but that's not enough to cause a tumor. And then the tumor can take several routes, either the BAP1 route, which is associated with specific genomic changes, or the class 1 route with SF3B1, AF1EX, and now adding PRAME to the mix, leading to higher metastasis rate in class 1s and a little higher in class 2 as well. And we drill down in this in a very detailed manner in a recent Nature Communications paper looking at the evolution of all these tumors. We also have recently launched the largest prospective clinical trial in uveal melanoma, with Bascom Palmer in our lab being the lead center, which includes 30 centers, where we prognostically want to validate PRAME and look for a couple of other things in those tumors as well. So we're very excited about discovering PRAME in uveal melanoma. And this is for several reasons. First, PRAME is overexpressed, meaning there's more of it. Anytime there's something more of it or activated in a cell, it's more easy to inhibit than if something's shut down. If something's shut down, like BAP1, it's very hard chemically to reactivate it again. Secondly, PRAME is only expressed in testis in big amounts, meaning if we find a therapy, potentially the side effects could be very low. Third, it was discovered in other melanomas as an antigen recognized by T cells, meaning the immune system in principle can detect high PRAME levels. And it's also expressed in many other cancer types, which means it's a major mechanism. It's not specific for uveal melanoma. There's a lot of interest in the research community. There's a lot of funding going into this for other cancers. And there's several clinical trials already looking at how can we target high PRAME expression that could be applicable for uveal melanoma as well. But the first question we had to ask is, does it really do something for uveal melanoma, or is it just a biomarker? Does it just sit there and indicate higher metastasis risk, but really the tumor doesn't care whether it's there or not? What you see here is another mouse model we have where we can inject the uveal melanoma cells in the eye, where they usually reside, and we add something to the cells that make them glow. And if you use a very specific, expensive camera, you can detect this glowing in a live animal. And you see the tumor has developed here in the eye. And then after a while, look at metastasis. You see the control animals here develop metastasis in most cases. But if we take PRAME out of the cells, most of the animals don't develop metastasis anymore. So yes, PRAME is actually doing something in those tumors, and targeting PRAME could be very beneficial for treating that tumor. Here another example of the liver, where the control mouse has some metastasis and the PRAME knockout does not. One interesting thing is that if we reintroduce PRAME into the cells, it really takes a while for them to change. So it takes for them 4 weeks before we couldn't detect anything really significant. But after that long time span, we see a lot of changes. And we see a lot of changes that make sense. We see that cell cycle is differentially regulated. We see genes that are involved in invasion are up, in tumor progression. So all that makes sense. And so this is the last research slide I'm going to show you. And this is what we think PRAME is doing and how we can target it and are currently working on. PRAME is part of this complex that puts little ubiquitin groups on proteins. And when PRAME is there in a cell, then these proteins that shouldn't be ubiquitinated are now ubiquitinated, which leads to their degradation, which then leads to all these things we see involved in cell cycle, invasion, and tumor progression. Now, the good news about this is that every step of this slide I show you is a point where we can intervene. We can block this interaction, we can counteract this ubiquitination, or target the effects directly. So this is the third big branch therapy involved, which we're targeting, looking at how can we counteract the effects of PRAME. With that, I'd like to close this session and especially thank our funding agencies, in particular CureInsight and the Jack Odell and John Dagres Research Award. And I'd like to stress this point, while we're very fortunate to gotten big funding from the National Cancer Institute and the Department of Defense, it's really the big findings we had in the past were all made with these smaller grants, because these allow us to go out and be a little more risky and try things. And once we find something, we can go back and get the firepower to spend a lot of money and put some big effort in. So we're very thankful for that. I'd like to close with our lab, with Dr. Harber being the head, and then the senior scientists, myself and Jeff M. Kuznetsov, who's the cell wizard. He does all the cell cultures and knocking down BAP1 and all that stuff. We have a lot of bioinformaticians recently, Christina Decatur, our lab manager, and all the other people that have contributed to the data I've been showing you today. Thank you.